so this is the idea that we'd like to pursue, and we're taking a number of approaches to trying to essentially turn on NRL in cells that don't normally want to turn it on. And so I just want to briefly recap some of the points that I've made. These are sort of the three questions that I think we need to try to address and understand in order to better figure out how to treat this disease. One is, how do normal photoreceptors develop? How does this developmental process occur normally? Because as I said, our hypothesis is that in retinoblastoma, there is an abnormal disruption of this developmental process, that the cell gets hung up here because there's a roadblock placed in the way in its path to differentiation. And so the next question then is, what is the nature of that roadblock in retinoblastoma? And lastly, the question is, how can we possibly force the tumor to differentiate, to get around this roadblock and to differentiate so that it exits the cell cycle, is no longer proliferating? And so far, what we have found is that RB1 is required to turn on NRL. This seems to be the case. And so this has led to the concept that failure to turn on NRL may be an important step in the process of tumorigenesis. And so then, in turn, that's led to the idea that you might possibly be able to treat these tumors by flipping NRL back on in the tumor. And there's a couple different ways one might be able to do that. One way is to use various genetic vehicles to deliver NRL to the tumor cells and to cause it to be expressed there. And other ways that we're pursuing is that we're screening panels of drugs that may have the properties that they could induce NRL expression in the tumor cells and thereby force them to differentiate. So the ultimate goal of all this, obviously, is to devise ways of treating these children non-invasively, that don't require surgery, that don't require removal of the eye, and it could potentially be used as an adjunct to other therapies like chemotherapy or radiation that might be less toxic and allow them to be treated so that you get two red reflexes of the eye instead of this asymmetric reflex of light. So with that, I just want to credit the people that I've been working with on this. One is a very talented postdoc in my laboratory, Dr. Stacy Donovan, who's been doing a lot of these experiments, and Dr. Bill Harbour, my collaborator in ophthalmology. And as I mentioned, of course, these studies are supported by the generous funding of the Children's Discovery Institute. And after I answer any questions that you might have, I brought my microscope along, which is sort of my daily workhorse in my clinical work. And it's a two-headed microscope, so it's basically a teaching microscope. This is how we teach our residents. And so I can look in one side, and any of you that wish can look in the other, and I'll show you an actual human eye that unfortunately had to be removed because of retinoblastoma. And you can see what this tumor looks like through the microscope and how we make that diagnosis. But if you don't want to look through the microscope, I also brought a nice big printed version of it. You can look at it. It's a little easier to point out, I think. So if you have any questions, I'd be happy to take them. Yes?
When the rods and the cones are formed, you said that's the end of the redevelop or regeneration process. So they don't regenerate through a person's life, or do they just, are they the same cells you had in the beginning?
This is a very interesting question, and one that other people in my lab are very interested in. It turns out that neurons that we're born with are the ones we have throughout life. And this is not just true in the retina, unfortunately, but it's true in other parts of the nervous system in the brain. And that's exactly why when you hear about degenerative diseases, most of them are neurodegenerative diseases. So diseases like Alzheimer's disease, Parkinson's disease, and others. And the reason for that is once those cells die, for whatever reason, neurons, they are not replaced, essentially. Okay? And so that is a real barrier to treatment. We could figure out ways of introducing cells to replace the cells that are lost, or coaxing adjacent cells to, say, proliferate and then redifferentiate, so regenerate. this would be phenomenal. It turns out that some lower organisms can actually regenerate very well. And so there are studies that were done a number of years ago in salamanders. And so you can take the retina out of a salamander, and after a few weeks, it will completely regenerate this retina and can actually see with it again. It's pretty remarkable. But for whatever reasons that are not entirely understood, as you move up the evolutionary scale to humans, the ability to regenerate has been lost.
But could you inject like an embryonic cell that has, not run its course yet, and have it adopt those features?
Exactly. So you're talking stem cell therapy, basically. And obviously there's a lot of interest in that. One problem that people are encountering with stem cell therapy, particularly in regard to the retina, is it's fairly easy to generate a stem cell. And nowadays they can even do it from some cells from fibroblasts in someone's skin. This is a big breakthrough that occurred about 4 years ago. So we can easily generate the stem cells. But the problem is going from the stem cell, let me go back to my own figure here. Going from the cell at the top here and causing it to go all the way down that path so that you can then reimplant it and restore the vision. That step is very hard. The reimplanting step is very hard as well. So, but a lot of people are working on that, and of course not only in the context of the retina, but in many other cell types. For example, trying to replace the cells that produce insulin in the pancreas in patients with diabetes. Yes?
Do you know what NRL does? And is there a way to sort of not necessarily try to turn the NRL on, but just use whatever product it might make or do whatever it does to get around it?
It's a very good question, too. NRL is what we call a transcription factor, and it's a protein that binds to different places in DNA. So it binds in many places across the genome, and itself turns on or turns off other genes. Okay, so its function would be very hard to replicate, say, with a drug or something like that, because you have to have a drug that knew precisely where to bind across the genome. And so, to my knowledge, I can't conceive of a way to do that. Maybe someone could, but I think the best way would be to get it itself to turn on. It's a very sophisticated and very complicated function that it has. Any other questions? Yes.
In your patient there, when you had the two pictures, one had white eye and one had a red eye, is that Photoshop or?
I hate to say it, but it was. So the goal would be that we don't have
thought, like,
Photoshop. Unfortunately, so that patient was cured, but she lost her eye. And so, they have to remove the eye, and after that, they have very good prosthesis they can put in, and you almost, just by glancing at a child that's had an eye removed, you wouldn't even know that they had an eye replaced, except the eye obviously doesn't see anymore. They have essentially a glass eye. Mm-hmm. So yeah, in her case, it had to be removed.
What happens if you don't detect it?
This is a problem. And that's why I said there's these deaths, but mostly in the developing world, where it's often not detected and too late, the tumor continues to grow. Okay. If you can catch the tumor before it's passed outside of the eye, then you can usually effect a cure. But once it's outside of the eye and passes out, and then perhaps gets into the bloodstream and passes around the body as a metastasis, then it becomes very difficult to treat. And so this often leads to the child's demise. But as I said, we're getting better and better at treating this, so when people pick it up, the patient gets treated promptly. If the tumor is not outside the eye, then often it can be cured.
Is that one of the only symptoms, or is, just
so
happening later?
The children are often young, sometimes still infants, so they might not be able to communicate that well. And so obviously if you have a big tumor in there like that, it's affecting your vision. But since a 2 or 3-year-old has another eye, they might not complain about that, or you might not even notice the effect on it. So they would have visual symptoms as well, but they're not always good at communicating that. And of course, if it becomes really advanced or starts progressing and spreading around the body, then there might be other symptoms.